You've seen that. I told the TAs I would like to have them back by tonight, so I'm hopeful for tomorrow. Of course, I'll put out an email letting you know when they are available, and they will be available in the BB office at that time. So what do you guys think about the exam? Easier than last exam? Harder than last exam? Oh. That goes to show you how much I know. I would have guessed that you would have said easier. You thought so? Okay. So, thoughts or perspectives on it? Any thoughts about it? Everybody gets A's? If everybody got A's, then your grade would have no value. Would you want that? Okay. I mean, the value of an A would just go like that. You would say, oh, you know, I don't want this, right? I want an A++ or something, right? Everybody would want that. Then everybody would want an A++.
Yeah, I'll take it.
Okay. Well, we're rapidly moving to the end of the term, obviously. And so we have a lot of things to do, but we're very much on target for where we need to be. We finished exactly last exam where I thought we would, so that's very good. We're getting ready to talk now in some detail about molecular processes, including transcription and translation. And as you saw with DNA replication, some of this material will be new. Some of this will be review for some of you. And hopefully everybody gets something very new and useful out of what I have to say. So that's important. It surprises me and disappoints me the number of times I hear students confuse transcription and translation. Okay? If there's one thing that I can assure you you're going to know, need to know in this class, it's the difference in those two terms. Well, transcription makes RNA. All right. Remember that. Okay? You'll be amazed at how quickly you will forget that on an exam. Translation makes protein, transcription makes RNA, and it uses DNA as a template. Okay? Transcription is a very elaborate and very orchestrated process. In eukaryotic cells, I would say it is very much orchestrated much more elaborately than it is in prokaryotic cells. We're going to see in prokaryotic cells that the controls are relatively simple compared to eukaryotic cells, but nonetheless there are a series of events that happen. Transcription I'll be talking about will occur at two levels. So today and tomorrow I'll be talking about fairly general mechanisms of transcription. Okay? What happens in the initiation, elongation, termination phases of transcription. These are very general sorts of things. I'll also say a few words about eukaryotic transcription. I may get into that today. I may not get into it until Wednesday. But then the real nuts and bolts that we'll talk about transcription will happen later. Your book actually splits, and it's kind of an interesting split, splits transcription into general transcription, which is what we're talking about here, and then more involved levels of what's called gene expression, of which transcription is a component of that. So we will finish general transcription on Wednesday, and then sometime next week, we'll actually go through gene expression. And so gene expression will give us some more detail about these various processes. Well, transcription, as I said, is the process whereby DNA information is converted into RNA. And one of the common questions I get is, well, why does that happen? Why have something like RNA? As we will see in talking about eukaryotes, that's absolutely essential, and the reason that it's essential is because transcription and translation occur at different places in the cell. Transcription occurs in the nucleus, and translation occurs out in the cytoplasm. So we have to have something that carries that information from the place where the RNA is being made or the nucleic acid is being made to the place where it's actually translated. There are other reasons for doing this, and as we shall see, one of the things that happens with RNA is because it is an intermediate as such, it's disposable. And that's important. Okay? It's disposable. Cells break down RNA. It's important that they do that because cells don't want to be making a protein forever. Just because you've made an RNA doesn't mean you want to be making that protein all the time. Cells are efficient. Cells have to make things when they need them and not make them when they don't need them. If you had an RNA sitting around that was always available to make protein, cells would be making certain proteins at times that would not be beneficial to them. So having essentially a disposable nucleic acid like RNA meets the cellular needs very, very well. Okay. Well, not surprisingly, RNA polymerase is the enzyme that catalyzes the phosphodiester bonds necessary to make RNA. RNA polymerases act like DNA polymerases in, quote, reading a strand and then putting in complementary base pairs, but instead of making a DNA strand, it makes an RNA strand. Now you will remember, hopefully, when I talked about DNA polymerases, I said they were shaped a bit like a hand. And we had that hand where the DNA double helix fit inside of here. And not surprisingly, we look at the structure of RNA polymerases, and we see they too have a shape that is not unlike that of a hand. Okay? One of the things that you learn in biochemistry and you get reinforced in biophysics, if you ever take biophysics, is that structure implies function. Okay? So the structure here is absolutely essential for its function. And when you compare two different RNA polymerases, one from eukaryotic cells and one from prokaryotic cells, again, it's not surprising that we see similar structure because they have similar functions. Now, there are differences in transcription between prokaryotes and eukaryotes, but in both cases, the aim is making RNA. So making RNA is critical. One of the things that we see in making RNA that's very different from DNA is, first of all, we have very specific places in the genome where RNA is being made, and cells have some very interesting and elaborate controls for recognizing, first of all, where those places are, when to make RNA, and how much RNA to make. Second thing that we see is that transcription occurs a lot less rapidly than does DNA replication. Okay? We saw in prokaryotic cells, for example, that DNA replication was occurring at 1,000 nucleotides a second. In prokaryotic cells, the very same cells where DNA replication is going 1,000 nucleotides a second, we see transcription occurring at the rate of about 50 nucleotides a second. So it's going considerably slower than is DNA replication, and that's probably because A, it's not optimized, and B, it doesn't make the same type of replication fork that a DNA polymerase does. Now, RNA polymerase, and what we're seeing here, in fact, most of what I'm going to be talking about today actually relates to prokaryotic cells. RNA polymerase is a multi-subunit protein. You can see some of the various subunits here. There are a total of 5 proteins, including 2 copies of alpha subunit inside the protein. We're not going to worry about specific functions of these first 3, but we will talk some about the sigma factor. And the sigma factor turns out to be a very interesting protein in the process of the initiation of the phenomenon of transcription. Okay? Now, prokaryotic RNA polymerase, as I said, has 5 subunits. There is only one RNA polymerase in prokaryotic cells. The RNA polymerase in prokaryotic cells makes all of the different types of RNA. So, of course, we see the major types of RNA that include transfer RNA, messenger RNA, and ribosomal RNA. All 3 of those RNAs are made by the single prokaryotic RNA polymerase. Eukaryotes have specialized RNA polymerases, and they actually have 3 major ones that are involved in making the 3 major types of RNA, and I'll say more about those as I get further along. Okay. The active site of the RNA polymerase, I didn't talk about this for DNA polymerase, and I'm not going to talk really much about it for RNA polymerase either, but they use a similar mechanism of making the phosphodiester bonds. Remember that an RNA polymerase is making phosphodiester bonds. You can see that when the RNA polymerase lays down, as it were, on a piece of DNA, it's big enough to span about 35 to 40 nucleotides on a piece of DNA. So this region of 35 to 40 nucleotides we'll see actually is important for some signals, as it were, that are contained within the DNA molecule. All right? Now you see some numbers up here. You see minus 20, minus 30, minus 40. And it turns out, Gesundheit, it turns out that transcription, the place where transcription starts for a given gene, the start site has a numbering of 0. So 0 here, there's minus 10. 0 will be about right here where the actual transcription starts. And everything in front of that 0 has a minus number. So minus 10 means we're 10 nucleotides away from the place where the RNA starts to be made. Obviously, minus 40 is about 40 nucleotides ahead of where that goes. Now, like DNA polymerase, RNA polymerase works only in the 5' to 3' direction. That is, RNA is made exclusively in the 5' to 3' direction. Okay? It's like DNA replication, it's one of the few absolutes in biology. When we say always, we mean always. There are no exceptions. RNA is made only in the 5' to 3' direction. Well, we've got two strands here. All right? The cell has to understand which strand is the one to copy and which one not to copy. Okay? So what we have then is something I will show you here in just a minute about the naming of those strands. Before I do that, however, I want to describe a technique that we use to understand where it is that transcription starts. Where does transcription start? How do we know? All right? Well, there's a lot of different experiments that one can do to determine that. But let's say I had a piece of DNA, and I think that there might be a place in that DNA that transcription would start. How would I determine where that start site is? Well, what this experiment shows is a way of studying that. Okay? Well, one of the things that we're interested in when we start transcription, you saw in the last slide how the RNA polymerase bound to the DNA at a specific site. So this technique that I'm showing you actually tells us where a protein has bound and covered up DNA. Has bound and covered up DNA. So let me tell you how we do this. We take a strand of DNA. All right? And that strand of DNA we label at one end. We can call this the 3' end if you want to up here. And that labeling involves putting a little radioactive phosphorus out there at that end. Okay? The radioactivity allows us to follow, essentially, what happens to this molecule. I take this labeled fragment, and this fragment, somewhere in it, has a place that I think that DNA binds to. The technique I'm describing to you is called footprinting. And footprinting is very, very valuable for, again, telling us where a protein binds specifically to a DNA. In my experiment, I take that labeled DNA molecule, and I divide it in half. Half of it goes to this set of reactions, and half of it goes to this set of reactions. The reactions that this fragment goes under are identical in each case. The difference being, in the reactions that occur on the right, I put my protein that binds to that region. It might be an RNA polymerase. It might be some other protein that I think binds in that place. The other reactions don't have that protein. Well, what are the reactions that I run? The reactions that I run involve treating the labeled DNA fragments with a nuclease. Okay? Now, a nuclease is an enzyme that chews up DNA or RNA. And this particular nuclease that we use is called DNase, D-N-A-S-E. DNase will nick DNA. It'll break one phosphodiester bond. That's how it works. If I put a ton of DNase in a reaction, it will break every phosphodiester bond in that molecule, and I won't get any information out. Okay? I'm interested in understanding where a protein binds, and so in order to do this, I have to use a small enough quantity of DNase such that on average, each starting fragment gets nicked one time. Okay? So I use a fairly dilute mixture of DNA so that on average each fragment gets nicked one time. Well, where does it nick? It turns out DNA nicks randomly. So if I look solely over here at these to start with, I say, well, if it nicks randomly and it hits each one time, notice I've separated the double strand into single strands by here. Okay, so I've gotten rid of the unlabeled strand up at this point. The unlabeled strand is gone. All I'm looking at is the labeled strand. If DNA is nicked randomly, then I should get a random mixture of every possible size of nucleotide. Okay? Some might be 50 nucleotides long, some 49, some 48, some 47, et cetera, all the way down to a single nucleotide. Everybody understand that? So I've got a small quantity now of that DNase that I'm using. Well, if I compare the DNase nicking with in the two that has no protein with the one that has the protein that binds to my sequence, I will see something interesting happen. The protein that binds to my sequence recognizes that specific sequence. And it covers it up. It grabs a hold of it. That's what a protein does when it binds to DNA. It grabs a hold of it and it covers it up. The places where it covers it up protects that DNA from getting chewed up by a nuclease. So DNase can't touch any of the sequences underneath this yellow blob. Everybody with me? Okay. So when I compare these two, I see, well, the guy can nick here. It can't nick in here, but it can nick in here. If I take this mixture and I take this mixture and I separate the fragments on a gel, I see something interesting. For the mixture on the left, I see what we call a ladder. It looks like a ladder. This one might have one nucleotide, 2, 3, 4, 5, 6, 7, 8, all the way up to, say, a couple of hundred. Each one differing by one nucleotide because there's nothing stopping the nuclease from cutting over here. Over here, wherever the protein was sitting, those sizes will not have nicks because the protein was covering it up. So I could look at this, for example, and say if this was 1, 2, 3, I would say this protein was covering up spaces 4, 5, and 6 on that DNA molecule. I therefore know where that protein is binding to DNA, and if this were an RNA polymerase, for example, I would know approximately, or pretty close, where that transcriptional start site would be. Okay, does that make sense? Yes? How do I stop this guy from making RNA? Is that the question? Okay. So how would I put this in here and not allow transcription to occur? How do I make it stay right there? It's very easy. I don't put any ribonucleotides in there. So without putting any ribonucleotides in there, there's nothing for the polymerase to make RNA and elongate. It's a good question. It's sitting there waiting for ribonucleotides. Pretty cool, huh? Can I put formaldehyde into it? Formaldehyde, well, what he's talking about is can I cross-link this protein to the DNA, and I can. But in this case, I don't even need to. Okay? There are some studies where I will want to do that, but I don't need to in this case. Yeah. Other questions? Good questions. You guys can explain this to me? Good. Good to hear. Good to hear on a Monday. All right. All right, so footprinting is a very valuable tool. Well, thanks to footprinting, we can tell where those start sequences are. Now, let's actually look at some sequences in DNA that people have analyzed once they know where the start sequence is for a whole bunch of different genes. When they look at this, they discover something very interesting. Here is transcriptional start site. Here is essentially 0. We look in a region called the -10 region. And by the way, this is true in prokaryotic cells. Eukaryotic cells have a similar sequence, but it's a little further away, and I'll talk about that later. So all I'm talking about right now is prokaryotic cells. When I look at the -10 region of prokaryotic cells, I discover something interesting. The sequences are very similar, not identical, but very similar to each other. And if we look at that, TAT, AAT. Okay, we call that a Tata box. All right? You find that funny? A Tata box derives its name from the fact that it's rich in A, T, and TA. All right? Now, if we think back to the chemical nature of AT base pairs compared to GC, what did we notice? Fewer hydrogen bonds, right? And fewer hydrogen bonds meant it was easier to pull apart AT base pairs than it was GC base pairs. How convenient, as they say. Okay? Cells aren't stupid. Okay? They have this control region that is rich in AT base pairs. And so like we saw with DNA replication, where we had those AT base pairs rich and it was easier to separate the strands so we could start replication, similarly we see a -10 sequence that is AT rich that makes it easier to start transcription. Now, you notice that not all of these sequences are identical. Okay? TAT, GTT. It's similar, but it's not the same. TTT, CAT. Okay? So we see some variation in there. We wonder what that variation means. And I can tell you. All right? We know in prokaryotic cells that the closer this sequence, this -10 sequence, is to the consensus sequence, the more transcription occurs. The closer the -10 sequence is to the consensus sequence, more transcription occurs. We refer to this as a strong promoter. And now I've just introduced a new term to you. A promoter is a control region in DNA for starting transcription. A promoter is a control region in DNA for starting transcription. Some promoters are what we call strong, meaning that a lot of transcription happens off of them. And some promoters are very weak. Very little transcription happens. Well, this makes a lot of sense. Some proteins in the cell we've got to have a lot of. All right? The proteins that make up the ribosome, for example. If we don't have enough proteins to do translation, we're in trouble. So cells make a ton of proteins that assemble into ribosomes. Okay? When I talked about DNA polymerase III, I didn't tell you, but I'll tell you now. In an entire E. coli cell, there's only 5 or 6 DNA polymerase IIIs. Only 5 or 6. Why so few? Well, remember, 2 of them start here and go on and don't come off. The cell doesn't need any more than that. Okay? So the cell is wasting energy if it's making a whole bunch of DNA polymerase III because it just doesn't need that many. Okay? Well, we could imagine that the promoter for DNA polymerase III would probably not look very similar to this because I told you the closer it was to this, the more transcription occurred off of that sequence. Okay? Everybody with me? Okay. There's our Tata box. All right. You like that, huh? All right. There are other sequences that appear to influence transcription in prokaryotes as well. There's another sequence that doesn't have such an exciting name. It's called the -35 sequence, and it's located approximately 35 nucleotides away. Now, if you think about the dimensions of that DNA that the RNA polymerase was sitting on, it stretched back just about as far as the -35. We can think of this sequence and this sequence as combining to influence how strong transcription is, how much RNA is made. Again, we see similarities. We don't see identity in every case with this one. Okay. Now, eukaryotic cells, I'll talk more about next time. Eukaryotic cells have many control elements. Eukaryotic cells have more genes. They have a lot more complexity. They have a lot larger chromosome. And so they have a lot more elaborate controls as well. Well, this figure illustrates some of the differences that we can see in different promoters. If we look at the heat shock promoter, for example, we see, wow, that guy is pretty different from the Tata box. doesn't look very much like it. Right? How does a cell control this? Is it just simply a matter of some of them are used a lot and others aren't used so much? The answer is no. Okay? We've talked about the importance of cells being able to respond to their environment. Last term we talked about it with respect to metabolism. Now we're going to talk about it with respect to which genes to make and how much of them to make. Okay? Well, let's talk about heat shock genes. Okay? Heat shock genes are pretty cool genes. They are genes that help a protein to fold properly. You learned last term the importance of the proper folding of proteins for proper protein function. And one of the things you learned about proper protein folding was the fact that if you misfold a protein or you unfold a protein, you can lose function. In some cases, you might make something like mad cow disease. So having your proteins properly folded is very important and very useful. Cells, especially bacterial cells, don't have a lot of control over the environments in which they exist. In our cells, we're pretty much at the same constant temperature, but even with a fever, our temperature rises a bit. And changes in temperature can, in fact, change the folding of a protein. If I'm a bacterial cell and I encounter a fairly warm environment, I'm going to unfold some of my proteins. If I have a mechanism of refolding those proteins properly, I have a better chance of survival in the long term than if I don't. So one of the strategies cells must have in responding to their environment is if they recognize that they've been at a higher temperature, inducing the production of proteins that help to fold proteins properly is a survival mechanism. Okay? Well, it means that my needs for those proteins are going to vary. If I'm sitting here in Kevin's gut, okay, after Kevin has just eaten a big juicy steak and doesn't have a fever, at 37 degrees, everything is fine and dandy. Kevin gets a fever and his temperature rises a few degrees. The bacterium doesn't like this. It's got to respond to that. It's got to make something to make that protein fold properly. Okay? All right, so that's what heat shock proteins do. Now I want to tell you how the cell controls that. Because all I've told you so far is that the only difference is the sequence difference. This wouldn't be made very much. But there would be times we would like to make a lot of this, or at least the E. coli cell would, because it's been exposed to a higher temperature. Well, how does the cell respond to that? And it turns out that in bacterial cells, the response is mediated by other proteins. Higher cells use other proteins as well. In bacteria, the responses are relatively simple. It comes down to that sigma factor that I talked about in the RNA polymerase. One of the ways that cells control which promoter to use and how much RNA to make from it comes from different sigma factors. That tells you that there are different ones that are made at different times. Okay? So if the cell has, for example, a sigma factor that is made most of the time dealing with 37 degrees and everybody is fine and dandy, then the sigma factor that's with that RNA polymerase does a really good job of recognizing standard promoters and making those genes because everybody's fine and dandy. Everybody's happy. Heat shock happens. E. coli starts making a different sigma factor. This different sigma factor now recognizes this type of sequence much more than the one we saw up here. As a result, the E. coli cell will start making a lot more heat shock proteins, and as a consequence, be able to respond to that environmental challenge much better. Cells have the ability to respond to heat. They have the ability to respond to heavy metals. Okay? I'm not talking about music here. Right? They have the ability to respond to heavy metals in a similar fashion. Heavy metals can be very poisonous to cells. So being able to make RNA for specific genes of interest according to the challenges that cells have is a very, very valuable thing. Now, sigma factor, as I said, plays a role in the initiation of transcription. Initiation is critical. If we don't start transcribing a gene, of course, we're not going to make the RNA that we need. Here's one for nitrogen starvation. We see something like this. Okay, questions on that? Yes? And means anything. So that could be any nucleotide at that point. Other questions? You guys are a dead group today. Too much studying? All right, let's talk about the actual process of making RNA. Okay? I've already alluded to the fact that in order to make RNA, strands have to be pulled apart. That's not surprising. We saw strands had to be pulled apart to make DNA. RNA polymerase has to pull strands apart. And you saw with that -10 sequence being TA rich that it's not surprising that unwinding will happen before the place where transcription actually starts. Okay? When we look at this, we see, for example, that we're making phosphodiester bonds just like we saw with DNA. We see that the starting materials are triphosphates, and we split off a pyrophosphate just like we saw with DNTPs in making DNA. It's going 5' to 3'. It's recognizing the base in the other strand to determine what to put in. The difference being that RNA contains uracil instead of thymine, and U pairs with A just like T pairs with A. Okay? Now, one other thing about RNA polymerases I didn't tell you that differs from DNA polymerases, we mentioned it when we talked about primase, is that RNA polymerases do not require a primer. No primers are necessary for making RNA starting with RNA polymerase. That's a real advantage in terms of simplicity for our way of thinking. All right, let's look at the synthesis of RNA in action. Okay? What you see here is an up-close and personal view of an RNA polymerase sitting on a DNA strand, and the RNA being synthesized is the green guy here. Interestingly, we see that as the RNA polymerase progresses, the RNA hangs off as a tail. Okay? It doesn't stay base paired with it for only just but a short stretch. All right? This tail, as we shall see, plays a role in the termination of transcription. It plays a role. Transcription has to have a specific place of starting, and as we shall see, transcription has a specific site for stopping. That's important because, again, cells don't want to make RNAs that are too long that aren't very useful to it. They want to have a place to start. For a short stretch, we see an RNA-DNA hybrid. That's what we see here. U paired with A, C paired with G, et cetera, et cetera. Okay? Now, there's two terms here that you should be aware of. First of all, the red strand is the one that's being copied. It's the one that the RNA polymerase is looking at and determining what bases to put in. That strand is called the template strand. So the strand that's being copied is called the template strand. The strand that is looped out and not being copied is called the coding strand. Well, why is it called the coding strand? Remember that in many cases we're making messenger RNA. So the messenger RNA is going to code for protein. The coding strand is going to have essentially the same sequence as the RNA, the difference being that U will be in its place instead of T. So it has the same sequence. Notice that the 5' end, which was the first thing that's being made, is hanging off, and the elongation is occurring at the 3' end. Okay. Now, something else you probably, you certainly can read, but you probably hadn't thought about is that, again, we are unwinding the duplex ahead of us. Now, it's not going at 6,000 RPM like we saw with the helicase. It's going much more slowly. Okay? But we do have some unwinding going on up front. What do you suppose we're going to need ahead of that unwinding? We're going to need a topoisomerase to keep things from getting knotted up. What you didn't think about was once we start putting things back together, this guy is underwound. we've got to put some tension back into it. So remember I said topoisomerases can unwind and also rewind? Back here, we have topoisomerases that are helping to rewind. That is, put more twists in. Okay, so now you can start to see that, again, when I talked previously about topoisomerases having specialized functions, that you can see that there might be a different topoisomerase back here than there is up here. Very specialized functions. The polymerase, in this case, is moving, as you note, from left to right, and that's the direction that transcription is occurring. Okay. I don't need to talk about that. Let's see. All right. Let's talk briefly. One of the things that happens is, this is actually bringing, I shouldn't have clicked on this slide because you're going to be looking at it instead of listening to me. But we've talked about the process of initiation. So let me back up to that, actually. Initiation is essential, as I said, and initiation is the way that transcription starts. Initiation has to involve recognition of the right places for things to start. And once things get started, we move to a phase called elongation. Now, elongation is relatively simple for us conceptually. After cells have made about 10 nucleotides, this transcription bubble complex that you saw changes. Okay? It changes. So I need to give you a couple terms about that before I go forward. So we've made this transcription bubble. We've got initiation started. We've got the strands pulled apart. That phase has a name. It's called the open complex. Okay? The open complex is literally the strands opened up and the RNA polymerase getting started. The open complex, okay, changes. After about 10 nucleotides, the change that occurs is the sigma factor says, okay, I've done my work, I'm leaving. The sigma factor disappears and goes out and helps start another transcription. The RNA polymerase, once it has been freed of the sigma factor, is in what we call the elongation phase, and it progresses along about 50 nucleotides a second, doing its thing, okay, until we get to the termination phase. Okay? So elongation happens, we get to the termination phase. I told you that the tail of the RNA plays a role in the termination phase. I want to tell you a little bit about that. Any questions? Yes, Shannon.
So you said one DNA or one RNA polymerase in the cell. Is that one type or one complex?
Yeah, one type. So there are many RNA polymerase molecules, but only one. They're all the same type. They're all identical.
And so is primase included in that?
Primase is not included in that. It's a very good question. So primase is not included in the RNA polymerases in terms of making RNA for. And the reason it's not included is it doesn't make an RNA that gets released. So primase makes an RNA, but that RNA is never released because it's chewed up by the DNA polymerase I.
Is it the same type of polymerase as the RNA that we're learning about, or is it a different structure?
Primase has a different structure than the RNA polymerase that we're talking about here. So, yeah. But good question. Other questions? Should we sing a song before we go forwards? Yes. Okay, I thought you guys would say that. All right. So, this song is a little bit ahead of where we are, but it's also a little bit behind where we are, so hopefully it's right in the middle of where we need to be. This one I get hoarse on, so I will hope that you will sing loudly so I won't get hoarse on it, okay? How many people like the Village People? Okay? I'm not going to go do all the gestures, but I will help you to sing the song. This is a song that I wrote several years ago called BDNA. And I want you to help me sing it loudly, okay? Let's let everybody outside know that we're in here. All right, here we go. To the tune of YMCA. Phosphates are in nucleotides, I say phosphates. Cover bases inside, I say phosphates. Span the 5 and 3 primes, there's no need to be real mixed up bases. Carry info, you see, I say bases. Are all complementary, I say bases. Like A, T, G, and C, they have got to be all paired up. Bow, 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 bow. It's fun to play with some BD. DNA. It's got a boatload of GCTA. It's got everything a polymerase needs when you melt all the A's and T's. It's fun to play with some bDNA. It's got a boatload of GCTA. You can make RNAs with a polymerase just by pairing up U's with A's. Proteins full of amino A's, I say proteins. Come with mRNAs, I say proteins. Require tRNAs, there is more. You need to translate codons. Like our friend UAC, I say codons. Come in clusters of 3, I say codons. Have one base wobble E. Now you can go forth and translate. Pow, 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 pow. It's fun to play with some bDNA. It's got a boatload of GCTA. With those hydrogen B's and right-hand helices, anti-parallel 5s and 3s. It's fun to play with some bDNA. It's got a boatload of GCTA. With those hydrogen B's and right-hand helices, anti-parallel 5s and 3s. One more time. It's fun to play with some bDNA. It's got a boatload of GCTA. With those hydrogen B's and right-hand helices, anti-parallel 5s and 3s. Okay, I made it through it. Woo! I should applaud you. That was very good singing. Wow.
Should do the hand.
What's that?
I have a
Question.
question. Can we sing happy birthday to you?
Anything but biochemistry, I suppose. Sure, go ahead. Please, go ahead. You have to stand up and lead it, okay?
Stand up. Okay.
So you get the A for today, right, Anjali? Okay. All right.
Happy birthday to you. Happy birthday to you. Happy birthday, dear Kevin. Happy birthday to you.
Thank you. Thank you. What's that? You want a speech? At my age, you don't look forward to them anymore, you know, so... But you're always happy that you've gotten at least one more year because the alternative is not so good. So thank you, thank you. You guys are the first class that ever did that to me. I will tell you that. I've been teaching for 15 years, so that's a nice surprise. Thank you. She gets an A for the day, so... No, she doesn't get an A for the day. Okay. Does that loosen everybody up a little bit so we can get back to biochemistry? No. We're not going to leave early today, no. Well, maybe we might leave a little bit early. Okay. All right. One of the ways that people study transcription, like the way they study other things, is with drugs. And I'm not talking about hallucinogens here either, okay? Drugs that stop specific processes, okay, allow us to understand how a process occurs. We saw last time we talked about how camptothecin or ciprofloxacin inhibited topoisomerase. By using those drugs, we could study their role in DNA replication. There are two drugs I want to mention that are useful in studying the process of transcription. One is called rifampicin. I'm jumping ahead, and I'll come back to the termination in a minute. Rifampicin has a very elaborate structure, as you can see here. It acts as an antibiotic because it inhibits the initiation phase of transcription. Okay? It inhibits the initiation phase of transcription, and it does it by binding to that very place in the hand where the DNA polymerase held the DNA. Once the DNA polymerase has gotten a hold of the DNA and has gotten started, rifampicin has no effect. However, if the rifampicin gets there before the DNA gets there, initiation of transcription is inhibited. Okay? So rifampicin is a very useful tool for studying the process of transcription. You can actually see its place right there. There it is in the middle of the hand, and that's where it's actually binding and stopping DNA from interacting with the polymerase. This is an RNA polymerase. Did I say DNA polymerase? Oh. Okay. You see what happens when I sing, there's no oxygen left in my brain. So, yes, RNA polymerase. Sorry. Okay. The other interesting compound, rifampicin works primarily on prokaryotic cells. It doesn't do very much in eukaryotic cells. However, the second compound, actinomycin D, has effects on both the initiation and elongation of both prokaryotic and eukaryotic cells. When we see something like that happening on a very broad spectrum and happening across processes, it tells us that there's something bigger that's involved. And this compound, actinomycin D, actually binds within a DNA helix. It disrupts the structure of the DNA, and the polymerase, the RNA polymerase, which I said correctly this time, the RNA polymerase doesn't move appropriately and is unable to copy the bases within it. So these two drugs are very, very useful for studying the process of transcription. Well, the last thing I want to say today relates to termination. And I'll do you a favor. I'll only talk about one of them. The second one I'll talk about at the beginning of next time. Okay? The one I want to talk about is really cool. Okay? It's a protein called Rho. Okay? RHO. Now, the analogy I like to give for Rho, remember that we've got this RNA that's hanging off of that transcription bubble. So we've got this long RNA that's now hanging off. This guy's gone along for a certain distance. Okay? Right? Rho is a protein that facilitates the stopping of the transcription. Okay? Now, let me go back here just a second. Rho's the one I want to talk about. Rho, actually, the one I showed I'll talk about next time. But Rho is a protein that is involved in stopping transcription. Okay? What does Rho do? How many people had to take gym class in high school and you had to climb a rope in the gymnasium? How many of you liked doing that? Yeah, you know, the first time I ever had to do it in gym, I hated it. But after I did it a couple times, it was kind of cool. I got there to look around, you know, and all this kind of stuff. Now, imagine, if you will, that you are climbing that rope, and instead of having a fixed rope, somebody at the top is letting it out as you're climbing it. Yeah, I mean, that'd be kind of... First of all, you hope that person's letting it out very gently, right? They don't drop you. All right? But imagine that you're climbing a rope that is coming down as you're climbing it up. That's what Rho's doing. RNA is being made, and Rho grabs onto the end of the RNA molecule and starts climbing it. Okay? It starts climbing it. What's happening is Rho is in a race with the RNA polymerase. Rho climbs the RNA faster than the RNA polymerase is moving through the DNA. Rho doesn't go on to start, obviously. It has to wait a while. But once Rho starts moving, it climbs that rope faster. Just like if you were climbing the rope faster in the gymnasium, faster than somebody was letting it out, you would eventually get to the top. Rho gets to the RNA polymerase, and what it does, it grabs a hold of the RNA polymerase, lifts its butt end up, and everything falls apart. Everything falls apart when that happens. Okay? Rho is a transcriptional terminator as a result of that. Well, how does it know where to stop, et cetera, et cetera? That's a little bit more involved than we're going to go into. But suffice it to say that RNA polymerases, as they're moving through a DNA sequence, some places they move faster, and some places they move slower. And guess where Rho catches the RNA polymerase? Where it's moving slower. Okay? So those sites tend to be pretty much common, and the transcriptional termination is pretty specific as a result of that. Okay, that's enough for today. We will stop there, and I will say more about termination on Wednesday. Hi. Yeah, what's up?
And I'm also a full-time employee at the veterinary college.
Yeah. Pull me up here.
My vacation or my time off from the semester is going to another clinic to work. It's actually the
Oh, uh-huh.
final group.
If you give me is somebody else in charge of this?
Okay.
Is somebody else in charge of this?
As far as my program? Or?
Yeah.
Yeah.
Just give me a note from whoever's in charge, and we can make arrangements. Yeah.
Okay.
Yeah.
All right.
I just need a note from somebody else.
Yep.
Okay. What's your name?